Hi, good afternoon, future fellow colleagues. And today is an asynchronous lecture on your module 2, which comprises chapters 3 to 5 of your Burton's Microbiology. This is Introduction to Microbes and Cellular Biology. So once again, I'm Dr. Beverly from our NetSci department. And for you to have a better understanding of medical microbiology, especially for your chosen career as future healthcare professionals, it is important that you have a very good foundation of microbiome. So this lecture will serve as complementary, will complement your studying through the entire Burton's book, at least for chapters 3 to 5. This lecture is not is not comprehensive. This is more like a summarized lecture, which wherein I will focus more on topics where I feel you have to emphasize your learning on. The uh, the nitpicking with the details, that's all up to you. That's where you reading the book comes in. Okay, so you need to listen to this lecture for you to be able to understand what you're reading. Okay, now I'm not sure how other professors would deal with um, teaching teaching nowadays, but in my personal opinion, I think that because you've never seen or experienced microbiology in person, at least in an academic setting, I would still want to hold your hand for a little bit to guide you as you tread through your journey through microbiome. But as I've said, this is not going to be comprehensive. These are just important landmarks in chapters 3 to 5 that I will be emphasizing. So you still need to read your book, and it's best that you have read chapters 3 to 5 already before you delve into this lecture. Okay, so it's very important that you take this subject seriously because you become a nurse one day. This is the basis of what it is that you do. You as a nurse, you don't just give drugs, medications, perform procedures without understanding the rationale why. You have to know why, and this is where it starts. Also, if you plan to proceed to pursue medicine in the future, this is the best framework for you, albeit it's not complete, but it will serve as a good foundation for your knowledge of microbiology. Okay, so my references for this lecture are, as I've said, Burton's Microbiology, which I've already shared in the Telegram group, a copy of the PDF file, illegal. Okay, and YouTube, of course. I got some videos from Crash Course YouTube channel and Professor Dave Explains. So if you are having a hard time trying to understand whatever it is that you're reading about in your books, YouTube is the one for me is one of the best ways to learn anything. We're missing out on the kinesthetic part of learning, but at least we're maximizing your visual and your auditory learning. So, okay, so let's start. Okay, so let's review some words that we have to learn in microbiome. Okay, so first is the cell. I'm pretty sure you're not alien to this anymore. The cell is the fundamental unit of any living organisms. Your cell obtains nutrients, from its environment to produce energy. Cytology is the term to denote study of the structure and function of cells. Metabolism is where chemical reactions that occur within a cell that allows it to grow and to reproduce. We are all capable of metabolism. Okay, so we are constantly in a state wherein simultaneous chemical reactions occur all at the same time that allows us to do whatever it is that we have to do every single day to survive. Your DNA, or your deoxyribonucleic acid, DNA, this is the genetic material. It makes up genes in its cell chromosome that allow the cell to adapt to its environment, usually by mutating. So your DNA is inside your chromosome. Your chromosome is like a gigantic bowl of genes in it, okay? So in your genes, you can find your DNA, which is the basic unit of your genetic material. So for our bodies to be able to mutate, we need our genes. And to mutate means that we can survive and thrive in whatever environmental situation there is. And it has allowed our bodies to survive for thousands of years, millions of years for our ancestor organisms, and it has not allowed us to become extinct yet. That's because we are constantly mutating. Okay, so bacterial cells exhibit all characteristics of life, albeit complex organelles are absent. Okay, so with bacterial cells, they're not as complex as our human cells, as animal cells, but they have the basic machinery for it to thrive. Now, viruses, on the other hand, they appear to be results of reverse evolution. That means they have very few genes and they don't have organelles. And they only contain a few genes, some few enzymes. It's pretty much it. No complex organelles whatsoever. They're actually pretty simple organisms. Even call them organisms, really. Now, as a review, your microbes, or the entirety of microorganisms that you're not seeing with the naked eye, can be divided into cellular, and cellular. So with your cellular, you have your viroids, your prions, and your viruses. Remember, your viroids and your viruses are the same. Okay. Now, these three things are cellular. I'll explain later what they are. Okay. Now, moving on to your mirror siya, hindi siya mirror galit. So with your cellular microorganisms, you have your prokaryotes and then your eukaryotes. Your prokaryotes, these are the organisms that belong to the Archaea, bacteria, cyanobacteria family. And your eukaryotes are your algae, your protozoa, your fungi. So we will go through all of that. Now, we have to compare the eukaryotic and the prokaryotic cell. Now, eukaryotic animal cells, as you can see, are pretty complex. No, this is already a review for you. I hope. I want you. There's a comprehensive detail of every cell organelle in a typical eukaryotic cell. Now, I want you to go through that again and understand it by heart, because you have to know that first before understanding what other variations of a cell can be seen in other families of microorganisms. Okay, so do I have to go through this? I feel like you know this already, but okay, we'll try to go through this one by one. Okay. Say something now. We'll start with your cell nucleus. Okay. So the cell nucleus is where all the genetic material is, diba? You have your nucleolus, it's the outside part, and then you have your nucleolus, which is the innermost part. That's where you can find all other proteins that's in the nucleus. But basically, all your genetic material is here in the nucleus. Okay. Now, your nucleus has a membrane. When you say a membrane, it's like a wall. It's not really a wall. It's more like a flabby barrier. Call it the nuclear membrane. Okay. And from your nuclear membrane here, you can see that you have these tunnels or labyrinthine structures here. These are your endoplasmic reticulum. No, so you have two types. You have the smooth endoplasmic reticulum, and then you have the rough endoplasmic reticulum. So your smooth endoplasmic reticulum usually is it's in charge of manufacturing fat lipids, whereas your rough endoplasmic reticulum here it facilitates protein synthesis, although it is not the organelle where protein synthesis itself occurs, right? But it facilitates. Okay? And then you have your SER, rough ER. The reason why your RER or your rough endoplasmic reticulum is rough is because of the presence of ribosomes. Your ribosomes are your protein factories, or they're basically proteins themselves, and they also allow production of protein. So I'm gonna sang RER, and that's what makes your endoplasmic reticulum rough. Now you have your Golgi apparatus. Your Golgi apparatus is more like your Ninja Van or your Maxim, your Kapandeliver. So it facilitates transport of uh, products from inside the cell to outside the cell. Okay, so may siyang courier. Okay. Of course, your mitochondrion, this little kidney-shaped things, no? I think you know already, this is the powerhouse of the cell. This is where all your energy generating, well, not all, but most of your energy generating activities take place. And without your mitochondrion, your cell will not be able to perform its function. So not all cells have a nucleus, no? Some of them do not have nucleus, but all of them have a mitochondria. And that's how important your mitochondria is. Okay, what else? You have your other you have your cytoplasm, of course. The cytoplasm is your cell body, and your cell membrane. Your cell membrane is, as I've said, it's not a wall, not rigid. Your cell membrane is though flabby and floppy. It's a semi-permeable membrane. That means it only chooses what it allows to go through. And for the other substances of it that wants to pass through the cell membrane, it has to go through certain toll gates. Toll gate, you have to a checkpoint. You have to have a certain characteristic for you to be able to get through, given that the mga required criteria are available, like i-check ang imo shape. You have to get into the correct alagyan. Mo na siya. Okay? Which you will discuss in your physiology class, hopefully. So you also have your accessory organelles, no? So you have your centrioles, your lysosomes. Your lysosomes are basically your cells' suicide bags. So when have you ever heard of the word SHTF? You look it up, what it is. So when SHTF occurs, your lysosomes will work into action and it will try to digest itself. So kung may mga foreign bodies that it needs to destroy, your lysosome, it's activated and it destroys everything, including yourself. Okay. So ano pa? Your peroxisomes are also accessory organelles, not that important. Not all cells have that. And then your villi and your cilia, whatever, these are for, sometimes your microvilli, these are not for movement. These are more like adherence or absorption. Your cilia, of course, is for locomotion or movement. Now, okay, we have discussed your eukaryotic cell, but now look at your prokaryotic cell. Look at how simple it is. It does have the basic components of your eukaryotic cell, but look at that, no? It's a cell wall, it's a cell membrane, it has a capsule, flagella, ribosome. So, very straightforward. Like, it doesn't have all the other stuff that the eukaryotic cells have. So, they're not as complex as eukaryotic cells. You can say it like that. Okay, sorry, may mga words na hagin screenshot. Wala na sasabihin. Okay, so, why is it that we do not have cell walls? Because cell walls can only be found in plants, algae, fungi, and most bacteria. And you cannot see cell wall in us animals, in protozoa, and mycoplasma species. Mycoplasma is actually a species of bacteria. Okay, so these are one of the few cell-lacking microorganisms that are bacteria. So that's mycoplasma. What's important about the cell wall? We'll discuss later. Now, I want you also to remember how you write your the names of your microorganisms. Huh? So, the example, let me try. For example, you have mycoplasma. No, so it's OMG, that is so ugly. Let me stop that. Sorry. I cannot erase that. Wait, sorry, may ari gusto ko technical. Okay, sorry. Okay, let's try again. So you have here your mycoplasma, no? That's the first name. And then the apelido, for example, is pneumonia. This is a legit bacteria, mycoplasma pneumonia. Now, when you write the names of your microorganisms, mycoplasma, italicized na siya dapat, ha? Or if it's not italicized, the first word, which is the genus, should be capitalized and the second word should not be. Okay, this is the species. The first word is the genus. So the genus should always have a capital in it. Okay, pneumonia. Pero usually it's italicized, no? So for example, in this case, mycoplasma here na italicized, you're referring to the genus of the bacteria. I cannot italicize in this thing. So, so you can also shorten that. You can write it as M. pneumoniae. Okay, ammonia siya, ha? You do not capitalize the species. You don't write it like that. Okay, you don't write it like that. So you have to write it as M. pneumoniae, something like that. Ammonia, ha? Short, dapat lowercase ang species, capitalized ang genus. Okay, so if it comes a time na you will have to write these names on your own, it has to be written correctly because I'll consider them wrong if you write them wrong. Okay. So again, a typical bacteria. Now we move on to a typical bacterial genome. Remember that eukaryotic cells, our DNA is a bit complex compared to a bacterial genome. When we say genome, this is referring to its gene makeup, no? So for example, sometimes your bacteria, its chromosome is separate from its plasmid. Hindi, wala tayong plasmid, guys, ha? Eukaryotic cells, we don't have plasmids. But bacteria, they have plasmid. This is how they, this is one of the ways where they can reproduce, kumbaga. Sorry. Reproduce their genetic material somewhere else. Okay. So again, your plasmid are not to be confused with plastid. Your plasmid are a small circular molecule of double-stranded DNA. Okay, so part niya siya sa genetic makeup of your bacteria, which is not part of the bacteria's chromosome. And however, there's also plastid, which is the site of photosynthesis in algae. So hindi ka mo ma-confuse ha sa spelling. Now, we move on to your bacterial cytoplasm. So your bacterial cytoplasm, which is basically just the the goo wherein the cell organelles can be found is what you divide as your cytoplasm. Now, in your bacteria, it's mostly ribosomes. Now, I want to introduce you to the Svedberg unit. Svedberg unit. Your Svedberg unit, amoy ni siya ang siyang S here, no? Now, your protein subunits are measured in Svedberg units. So, the ribosomes in your bacterial cytoplasm are made up of 7S or 70S or 70 Svedberg units. It's just a unit of measurement. Your 70 Svedberg units, ang ribosomes, are composed of 30S ribosomes and 50S ribosomes. You do not add them, ha? That's just what it is. Your 30 Svedberg units ribosomes and 50 Svedberg unit microsomes make up your 70 Svedberg units ribosomes. That's just what it is. Okay? Hindi na siya complex math. Amo lagi na siya yan. It's constant. Okay? Now, your bacterial cytoplasm can be visualized with stains in microscope. Okay, now we move on to your cell wall. Now, your bacterial cell wall, as we've said, no, most bacteria have cell wall. What is the significance of the cell wall other than they're just made that way? In microbiology, we utilize the bacteria's cell wall to identify the genus and species of the particular microorganism. So, for example, the presence of thick peptidoglycans, such as in your gram-positive organisms, will indicate that microorganism is most likely a gram-positive microorganism. Okay? So, explain na later, no na siyang mga gram-positive and gram-negative. Diba? As you've done your laboratory, or maybe you are still about to do it, sa inyong lab, you have the gram staining there. It will introduce you to gram staining. Although I will talk about it later here. Now, gram positive means nga it's a color blue, just a gram stain. Ang microorganism has an end color of blue, so it's gram positive. And that means that that gram positive organisms have thick peptidoglycan layer. Your peptidoglycan, its other word is murine. It's just polysaccharide chains. Let's say polysaccharide chains. These are multitudes of carbohydrate units. They become chains, no? And then they relate together by small protein chains. Or peptides are proteins. So, dako na siya mga carbohydrate nga subunits. It's multiple carbohydrate units na turn into one big polysaccharide, and then they become chains added in together by small peptide chains. So, dako na siya. Okay. So, kung gram-positive ang cell wall, thick ang niya peptidoglycan layer. Whereas, kung gram-negative siya, usually it's the color pink or red. Nipis ang iyang peptidoglycan layer, but it has an outer membrane. And this outer membrane is not present in gram-positive organisms. Okay. So, as I've said earlier, no, there's an exception to the rule. Your mycoplasma, sorry, it's not species, it's genus. The Mycoplasma genus of bacteria, they have no cell wall. At the same time, there are other cell wall deficient microorganisms. It's not just Mycoplasma. So when we identify bacteria, the first step usually to determine what it is we do a gram stain. Okay, not all bacteria are stainable by gram stain. Okay, mind you. So you would already have an idea what you're gonna do next. But the first identifying thing that we do is we do a gram stain, which is this is to stain the cell wall. So amo ni siya, no? Imagine that this is your peptidoglycan, and the specific things in here, these are your N-acetylmuramic acid and N-acetylglucosamine. These are your polysaccharides. And we'll not get into that, but I want you to at least visualize na thick cell wall in bacteria would kind of look like this. Okay. Pag-auto ka na. Now, this is the difference, no, between your gram-positive and your gram-negative. Remember that this green thing is your peptidoglycan. And as I've said earlier, with gram-positive organisms, microorganisms, cut down sa ilang peptidoglycan layer, no, dumaguba ganyan ni fix-it-feeding. Sorry, directed Ralph. Gubo niya ina yan. No? May maguba ina siya yan. Pero diri sa gram-negative nga mga bacterial cell wall, your peptidoglycan layer is thin. However, you have an outer membrane. So muna siya. And an inner membrane. Sa gram-positive damo lang gid siya peptidoglycan, may cell membrane siya pero isa lang. Compared sa gram-negative, na may outer siya kag may inner siya nga membrane. Okay, so muna siyang nag-help with staining. How it absorbs particular or how it has particular affinity to certain stains because of its cell wall makeup. Now, we move on to its glycocalyx. The glycocalyx is the slimy gelatinous material that is produced by the cell membrane and secreted outside the cell wall. So there are two types of glycocalyces. No? You have the slime layer. You can see this in your Pseudomonas species. Your slime layers are usually easily detachable nga glycocalyx. No? So it allows bacteria to glide or slide along surfaces, protects from certain antibiotics. Another type of glycocalyx is your capsule. You can see this in your Haemophilus influenzae. So this a capsule, a bacteria that has capsule in it or on it is called an encapsulated bacteria. And your H. influenzae is such. So your capsule can be polysaccharides combined with lipids and proteins, but your capsule is highly organized. It's attached firmly to the cell. Now, you cannot stain the capsule, but you can do negative stains. When you say negative stains, you stain the background such that you will not be able to, because you cannot stain the capsule, right? So what you do is you stain the background instead. And because you know what you're expecting for the background to have, anong color niya dapat, you can see that a particular layer in there nga wala stain should be the capsule. It's new. Okay. So, your capsule also serve antiphagocytic functions. What do you mean by antiphagocytic? When you say antiphagocytic, it allows the bacteria to survive without being destroyed by your white blood cells. So this is why some bacteria are just very resistant to certain antibiotics or even to our own immune system because of structures like your glycocalyx or your capsule. Later na. So, for example, no, another encapsulated microorganism is your Streptococcus pneumoniae. If you, this is an agar, okay? This is specifically a blood agar. How do we make an agar? As nurses, you don't have to do this. Pero when we were medtechs, no, partly ni siya sa amon yung microbiology class, we make the agars ourselves. Ideally, with the blood agar, you need sheep blood. In Pilipinas, tayo hindi ka makita sheep blood. Of course, kapag kami donor among ourselves. Hindi kami lang fleb. Tapos ibutang na naman ipod naman sa agar. So, may blood agar na kami. Anyway, so, when we try to grow bacteria on an agar, we call it inoculate. So, gina-inoculate mo, butangan mo siya. So, in this case, in this sample, this is your Streptococcus pneumoniae. Streptococcus pneumoniae is also an encapsulated bacteria. So, you would know nga capsule ni siya because look at that slimy thing there. Ew, it's so gross. Hindi na siya lumot, ha? That is Streptococcus pneumoniae's capsule. Okay? This is why only certain antibiotics can work with your Streptococcus pneumoniae because of the presence of a capsule. They're also hard to stain. So what you do is you do a negative stain. This is your negative staining, no? So you stain the background, and then you see areas here where it does not absorb the stain. That's the capsule of the microorganism. Very simple. Di ma-ship ka na medtech, pwede man. Joke lang. Abroad ka mo ya. Diba? Okay, next structure. Your bacteria also has flagella. Your flagella, these are protein appendages. When you say appendage, ano siya, attachment ni siya sa body. Like your limbs, your hands, your arms are your appendages. Okay, so these are protein appendages, and your flagella enable bacteria to move. So you have different, you know, a lot of your bacteria have different types of flagella. So a bacteria that has a flagella over their entire surface, they call them peritrichos. Peritrichos from the root word tricho or T-R-I-C-H, that's hair. That means it's Greek for hair. Okay, so when you see ang root word, you think of hair-like flagella. Okay. Next is your lophotrichos. This is a tuft of flagella on one end. So, may kumpol of flagella sa isa lang ka end. When you say amphitrichos, that's at each end. So, duwagid. When you say mono, that's mono, ha? Monotrichos, that's only one flagella on one end. Now, special consideration are your spirochetes. Spirochetes are spiral-shaped bacteria. One example of this is your syphilis. Syphilis is a spirochete. Okay, so your spirochetes, they have two flagella-like fibrils called axial filaments. And this allows your spirochetes to move in a helical manner. So, ang magsiling ka helical, kapag mo na. Okay. Social siya. Okay, so amo ni siya. For example, this is your salmonella. No, your salmonella is the causative agent for typhoid fever, for example. Kung mga kati ko lang pa, mga typhoid. Typhoid, can also cause dysentery. May mo na siya, no? Okay, so dysentery is basically you're just LBMing your guts out. So, your salmonella is a peritrichos na organism. So, na. Now, we also have flagella stains. No? So, flagella stains, these are stains na ang affiliate niya is a flagella na yung part. But we rarely do that in a Philippine setting because we're poor. Okay. Next is your pili or your fimbriae. Not pili like lupog, it's pili. Also, yung pili, not your pili, not it's pili. The singular term for pili is pilus. Okay. So, your pili or your fimbriae, it's not associated with motility. This is for adherence. Sometimes, it's also used for transfer of genetic material. And, you know, when your bacteria would perform sex with another bacteria, they call it conjugation. Okay. So, it's not really sex. I don't think it's pleasurable, but it's a transfer of genetic material. So, you know, when you have your partners, that's what you tell them. Do you want to transfer genetic material with me? Ew, gross. So, for example, this is your Proteus, no? Proteus vulgaris is also a bacteria. Your Proteus has a short, straight pili, no? Kadakong siya pili. Sometimes it's a flagella. But this one, this particular cell is trying to undergo binary fission. Pamutang tayo mga memes ako ng PowerPoint. Gusto na lang kung may time kung butang ako yung mga GIF. Okay, now, we move on to spores. Your bacteria, some of your bacteria has spores. If you play spores, the game, anyway, so spores in your bacteria are called endospores. Hindi siya spores sa plants, ha? Like spores, hindi siya yung like in bacteria spores, endospores. So this is a means of survival when nutrients in the environment is low. The formation of endospores is a process called sporulation. Spores allow your bacteria to be resistant to heat, dry, cold, and most chemicals. It allows your bacteria also to survive for many years in soil and dust. Some bacterial spores are resistant to boiling, heating, and some disinfectants. But you can also visualize them by staining. This is one of the challenges in treating bacterial infections, no? Because also of the presence of bacterial spores. So, for example, this is your Clostridium difficile. your Clostridium difficile is a causative agent also for dysentery also. Now, if you can see here, this is staining, ha? This is stained C. difficile. If you stain them, you can see that they have these darker ends, no? That's what you call, sorry, not the darker ends, the lighter end in the middle. That's your subterminal spore. Okay, so muna siyang sa spore niya. This is why your C. difficile is also very difficult to treat. No, so you have, for you to be able to, if you think about it, it's 2022, and we're supposed to be medically advanced in our treatment of bacteria and viruses, no? But think about it, even the simplest, or when I'm saying simple, like microorganisms that we've known for a long time can still be deadly. Your C. difficile is one of them. It's because of the presence of the spore. So the whole idea here is doctors should know what the causative agent of a particular disease is first to be able to treat it appropriately. Because, for example, your C. difficile it's not, di mo niya siya mabulong with your simple amoxicillin. You have to know what it is to be able to treat it properly. Because sometimes may mga presence of specialized characteristics like spores. Like, your C. difficile, it's not the only disease that causes diarrhea or dysentery. As I've told you, salmonella can produce dysentery, diarrhea, and so many other diseases. Okay, so that's why microbiology is important, because it saves lives. And that's why you have to learn it too, because you're going to be my future colleagues. Yeah, because you will save lives, diba? Come on, guys. Okay, so as a summary, you pause this video and then you take a screenshot and then you study this by heart. Okay. I will give you 5 seconds. Take a screenshot. Okay, na. Okay, next. Now, how do bacterial cells reproduce? Prokaryotic cells usually reproduce through binary fission. What is binary fission? Binary fission is when one parent cell divides into two daughter cells. Kanya mo divide, no? Now, there's this thing you call generation time. Generation time is the time it takes for a bacteria for binary fission to occur. Why is this important? Because of this particular table. Why is this table important? Because it is essential that we know when to treat our patients who have bacterial infections. We have to time the treatment appropriately. Okay, so knowing a bacteria's generation time would tell us when to treat. It's very easy, because if you give your antibiotics at the wrong time, so for example, your bacteria is already in the stationary phase, so suppose you can still treat it, pwede pa. But it's already dying, no? Or it's already approaching your decline phase. So dulo ko waste na lang of medications if you're going to treat in the stationary phase. However, the most or the best time to treat bacterial infections is during the exponential phase or the log phase, not the lag phase. This is the A and O here, they're very different. So the log phase is the time when your bacteria is still multiplying. Luga, ano pa siya, very, very mapusok, very excited yun siya nga magpadamo. So that's the best time to give your patients antibiotics during the exponential or the log phase. Okay. So remember this, you have the lag phase. Dili do ka, gaseto, focus environment. Okay, so ang bacteria do siya, duga ano pa siya, duga ay mo naman pag una naman yung environment, may na naman ko nutrients, OMG, hindi pa ko maka-reproduce. And then the moment your log phase starts, so muna na siya nga, ay hala ka namin, these nutrients, this is so amenable for my reproduction and for my invasion. So muna na siya. And then that's when you give your antibiotics. In the stationary phase, it's like, okay, nang saho-saho, naging ko diya, like, dili na lang gikuya go, pero hindi na ako magpadamo. Okay? So, and then it will approach death or decline. That's why it's important to time your bacteria so you can treat appropriately. Okay, so again, take a screenshot of this and then memorize this by heart. Now, how do you memorize this? This is easy because there's already a disease nga column, no? So, you know the causative agent of this particular disease. So, amo na siya. Once you're familiar with the disease process or the disease entity, it's very easy for you to memorize the causative agent. So, for example, Bacillus anthracis causes anthrax. You read up on anthrax and know its history and what it causes. Then you will remember Bacillus anthracis for the rest of your life. Okay? Chlamydophila pneumoniae. There's so many pneumonia subspecies. Tawag mo na sila nga genus, lain-lain na sila. They all cause pneumonia. Okay? So, Chlamydophila psittaci or psittacosis, or you get this from birds. Chlamydia trachomatis or trachoma, this is one of the more common chlamydia nga genus nga STD. Your botulinum, Clostridium botulinum, Clostridium tetani causes botulism and tetanus. What is the difference between your botulism and tetanus? You read that up para hapos siya i-memorize. Okay? Or you watch YouTube. No? Instead of being on TikTok, kag magsagay, kada scroll up, you YouTube this. Or you make a TikTok out of this. OMG, maximum learning, guys. Di ba mo itong kasi nyo, no? May pneumonia na mga subspecies, pero Klebsiella, ni siya yan. So, ano mas common sa aton? Ari ang Mycobacterium tuberculosis and your Mycobacterium leprae. Okay? Ano pa ginang common? Is your gonorrhea, causative agent for your gonorrhea. Okay? Nisimidinity this is also common in our local, no? It causes meningitis. Deadlier in children. And your strep pneumoniae here, Streptococcus pneumoniae, this is a very common causative agent for pneumonia. And then cholera, which is endemic in certain parts of the country. When you say endemic, no, it's a disease entity that's only present in a certain local. Compared to like a pandemic, kung silang ka-pandemic, that's a worldwide That's a worldwide nga state. But with Vibrio cholerae, that is it's endemic in certain areas, meaning sa certain locales lang. Also, in some cases, these bacteria cause more than one disease. So, for example, ano nga mga bacteria cause other disease? So far, madunduman ko. Next, you familiarize yourself with this. I'm not sure if this will come out in the exams, but at least familiarize. But if you're going to be nurses, these are good to know things. Good to know ni siya yan. Okay. No, sorry. Nice to know. Nice to know. Now, when you're trying to study bala, you have to sort ano ang imong important to know, good to know, and nice to know. Yung mga important to know, mo gini siya ang do very basic na will most likely come out in your boards in the future, or do ka even if you pursue medical school, magwa man na siya gyapon. That's very important to know or gagwa sa exams. You're good to know mga okay, sige, you still have to learn it. Pero these are the things that if you don't learn them, basi hindi ka kapasa. Like you have, like inabalang na-learn mo lang bituod ang mga very important to know, it's only enough to get you until 74. Pero if you know your good to knows, guaranteed 75. But if you're nice to know, that will get you up to 80, 85, 90. Pakauna siya. Okay, so muna siya. You sort your information para hapos mo siya ma-absorb. That will guarantee survival in nursing. Okay. Now we move on to your viruses. Your viruses, you have a fundamental unit of your virus is called the virion. Okay. So a typical virion would have a DNA or an RNA. So ang DNA or RNA, ang muna siyang iyang genetic material, no? So for example, nucleic acid. So siya, no? Muna siya. Parang pink. Tapos may protein coat na siya. That's the capsid. So amo na siyang iyang do suludlan. And then if you have your nucleic acid plus your capsid, that's what you call a nucleocapsid. So what your viruses, there's a debate whether you can even call them living organisms because not sure organelles. Some scientists would still say they're living organisms. Some would say they're not. But the whole point is they need to invade to take over to take over a functioning cell to produce new virions. Okay. So ano ni sila yung mga pyrate. So now I want you also to familiarize yourself with this. Okay. Dugay pa man in your boards. It's like three years from now. But this will probably come out in your boards. Kaya gagawa na siya sa medtech boards, medicine boards. But with you, I will have to ask my nurse ng mga family members kung gagawa na siya sa boards. But this is good to know. I would say this is good to know. Okay. Remember that your viruses are divided into, depending on their nucleic acids. So you have your DNA viruses and your RNA viruses. RNA is your ribonucleic acid. Your DNA is your deoxyribonucleic acid. So, for example, with your RNA viruses, you can still divide them whether they have an envelope or wala sila envelope. Umuni wala ho, may ara siya envelope, ining yellow, ni wala nga do may outside pag siya yung masulod lanon. So, amo na siya. So, may envelope siya wala, itong yellow, or is it non-envelope, amo to siya, ang wala siya ang yellow. So, kung non-envelope siya, is it single-stranded RNA? Because your RNA can be double-stranded. Your DNA is double-stranded, no? In humans, yeah, DNA is double-stranded. Your RNA in humans is single-stranded. But in viruses, because they're very weird, so sometimes they can be single-stranded. When you say positive sense, there is a direction wherein when your RNA, ano na siya mo galaba na siya. So the more, kung nag-multiply ang imong RNA, when you're saying multiply, like dugi na padamo, niya bilang niya genetic material, galaba na siya. So when it's following the proper direction, you call it positive sense. Kaya may proper direction na siya, guys. No, mabalaan nyo na siya eventually kung may mga lesson na kamo sa cytogenetics. But there is a proper kung sa atin, kung kita bala, magsulat kita nga mga Pinoy, magsulat taya, it's from left to right, or sorry, left to right, di ba? Whereas other nationalities, like ang mga Saudi, the way they would write is from right to left, di ba? So doon mo na siya. When you say positive sense, there it is following the default direction wherein your RNA material should be going. So single-stranded positive sense. So muna sa examples. Kung may ara ka naman, non-envelope double-stranded. So that's these examples. No? Your rotavirus is the most common cause of diarrhea in children. Okay, at least mo na lang nung dumon mo, rotavirus na lang. Tanan nga, most 95% ng lupot sa bata is not caused by a bacteria. It's by a virus called rotavirus. So gaayo lang siya. Okay, so don't panic kung lupot ang inyong mga manghod between 1 to 5 years old. Next, sa mga envelope mo naman nga RNA virus, yung envelope mo itong yellow by. They can be single-stranded positive sense, they can be single-stranded negative sense. Magsiling ka naman yung retrovirus, suli siya ya, no? So, wala siya go-follow nga from point A to point B to point C to point D. Siya ya, from point Z to point A. Suli siya ya. So, amo ni siya yung mga lentiviruses, oncoviruses, your causative agent for your HIV, AIDS mo bala, is a retrovirus. So, pag sama na siya, that's the whole point. Okay, so may linear, may circular, may segmented. You just familiarize yourself with it. Ang mga mas common, di ba? Parvoviridae, no? Adeno is a very common cause of your common cold, sipon. No? Wala din nabutang ang COVID. On next time. Okay, now, remember that your virus They have to tick every box here sa characteristic. So it has to have, you have to know kung anong yung genetic material. Is it made up of DNA or RNA? Ano ang shape ng capsid? Pila ang numbers ng capsomer? Ano kadako ang capsid? May envelope na bala? Anong i-host? Ano ang disease niya? Ano yung target cell? Ano yung immunologic properties? Just familiarize yourself with that. Probably memorize that. Okay? Mga good to know niya siya. Now, I want you to watch this video, guys. Ha?
Hey, it's Professor Dave. Let's talk about viruses.
He knows a lot about the science stuff. Professor Dave explains.
We now have a pretty good understanding of what's going on inside a cell, so we are ready to look at different kinds of unicellular organisms. But before we get to those, let's take a look at something a bit simpler, a virus. Contrary to popular belief, viruses are not alive. They are much smaller and simpler than single-celled organisms like bacteria, and they do not meet most of the criteria that biologists agree are required to call something alive. Living organisms must be able to perform metabolism, making energy from food. Viruses cannot. Life must be able to reproduce out of its own capacity. Viruses do not. They can be considered biologically inert. So viruses exist in a kind of gray area in between simple molecules and living organisms. So what's inside a virus exactly? In truth, a virus is pretty much just genetic material in a protein casing. There is no membrane, no organelles, not much of anything we are used to seeing in living creatures. Nevertheless, viruses reproduce by injecting their genetic material into a host cell, thereby hijacking the cellular machinery of the cell and forcing it to make copies of the virus instead of what the cell would normally be doing, sort of like pirates on the high seas, capturing a large vessel and taking command. Viruses were discovered in the late 19th century when examining certain diseases that afflicted plants. It was found that sap from the plant would transmit the disease, even though no bacteria were visible in the sap when examined with a microscope. And the sap would still transmit the disease, even when it was filtered by a process meant to remove any such bacteria. This meant that the agent responsible for transmitting the disease must be way smaller than a single bacterium. But this agent could not be cultivated in test tubes or petri dishes, so it must also be much simpler than a bacterium. Later, as we became able to examine viruses with more sophisticated techniques, we began to realize the structure of the virus, which comes in a number of forms. Some are rod-shaped or helical, like the tobacco mosaic virus. Some are icosahedral, like an adenovirus. Some have a membranous envelope covered with spikes, like the influenza virus. And some even look like weird little spiders. This is called a bacteriophage, and it's kind of like a rod-shaped and icosahedral virus combined, with some fiber tails. The thing they all have in common is that they carry their own genetic material, which could be double-stranded or single-stranded, and either DNA or RNA. This will typically be found as either a single linear molecule or a circular molecule. The protein shell that encloses the genetic material is called the capsid, which comes in different shapes for different viruses, and the capsid is made of smaller subunits called capsomeres. That's really all there is to the structure of a virus. So how exactly do they reproduce? As we said earlier, viruses hijack the machinery of a host cell. This is because they do not have ribosomes or the other components necessary to express genes, so they need a cell to do it for them. Certain viruses are able to infect certain kinds of cells, and this is due to the system of recognition that must occur between the two. In order to get inside the cell, a virus must be recognized by surface receptors on the cell, so there must be some specificity for these receptors to that particular virion. For this reason, many viruses are specific to only a small set of species, or even one individual species, and sometimes even a particular type of cell found within that individual species. Once this recognition occurs, the virus either injects its genetic material into the cell if it's a bacteriophage, or the virus can be brought inside the cell completely intact through endocytosis. Once inside, the virus disassembles and the viral DNA gets transcribed and translated by all the parts of the cell that are typically busy working for the cell itself. Once there are many copies of the viral DNA, the capsid proteins reassemble and form new viral particles, up to hundreds or even thousands of them, which then exit the cell. Sometimes this process can damage or destroy the host cell, so let's look at the different mechanisms viral replication can utilize for bacteriophages, as these are the best understood viruses. With the lytic cycle, the host cell is terminated at the end of the replicative cycle. This happens once many viruses have been generated, and the cell bursts open or lyses, releasing them to then go and infect other cells. Given the exponential nature of this process, just a few successive lytic cycles can destroy an entire bacterial population in a couple of hours. By contrast, with the lysogenic cycle, the host cell is not destroyed. This is because rather than usurping the cellular machinery to exclusively produce viruses, the viral DNA is incorporated into the genome of the cell. We can refer to this kind of viral DNA as a prophage. This DNA remains largely silent, and the cell is able to divide many times, with each daughter cell also containing the prophage. Then some environmental signal may trigger a switch from the lysogenic mode to the lytic mode, where the prophage returns to the form of a separate circular DNA molecule, and all of the infected cells could lyse at once. Apart from bacteriophages, other viruses have envelopes, which allow them to enter and exit the cell by endocytosis and exocytosis without destroying the cell. So it is of great importance to the virus that it will be recognized by these surface receptors. Other viruses are considered retroviruses because they contain an enzyme called reverse transcriptase, which transcribes an RNA template into DNA, which is the opposite of normal transcription. There are even smaller infectious agents called viroids, which are naked circular RNA molecules that disrupt certain regulatory systems in plants, and prions, which have no genome but are instead infectious protein particles that cause other proteins in brain cells to aggregate and bring on disease symptoms, possibly including Alzheimer's and Parkinson's disease. It would seem that no cells are safe from viruses, but nature is clever and bacteria are constantly evolving. Chance mutations in genes that code for surface receptor proteins may result in receptors that no longer recognize a particular virus, so it can no longer enter the cell. Viruses in turn mutate at random, and if glycoproteins on a viral envelope become modified such that they will be recognized by the new receptors, they will proliferate anew. In this way, bacteria and viruses are engaged in constant evolutionary flux. The origin of viruses is still disputed, though it is generally thought that viruses came about shortly after unicellular life first evolved, And there are a number of anomalous viruses that contain up to several thousand genes, including some previously found only in cellular genomes. A discussion of the specific diseases caused by viruses and the strategies we use to combat them will have to wait for a pathology course at a later time. For now, let's move on to the biological structure of the simplest organisms. Thanks for watching, guys. Subscribe to my channel for more tutorials. Support me on Patreon so I can keep making content. And as always, feel free to email me, ProfessorDaveExplains@gmail.com.
So when I say you support Professor Dave, he did a better job at explaining than I did.
Hey, it's Professor Dave. Let's talk about viruses.
Okay, so take a pause this video, take a screenshot, and familiarize this by heart. Okay, so we'll discuss maybe a little bit. No, so are you familiar with what they are and anong disease entities that they cause? So hapo siya ma-memorize ninyo. Okay. So I have a cattail that's smashing my keyboard right now. Anyway, so for example, you have your pox viruses. Again, this is a double-stranded DNA with envelope. So anong ginakose ni? You have your variola virus that causes smallpox and your vaccinia virus that causes cowpox. These things have been eradicated from humans because of vaccines. So you also have your warts caused by your papillomavirus, no? And then your some tumors and cancers are caused by your polyomavirus, which are polyoma or papillomaviruses. No? Your cervical cancer is papillomavirus. Next is your herpes virus, obviously causes herpes. You have your shingles, your chicken pox, your herpes, genital herpes, and then you also have your cold sores. That's caused by a herpes virus. Pero herpes simplex 1, not the genital herpes as an STD. That's herpes simplex 2. Your sulalab is caused by your varicella, which is a herpes virus. What else? Ang tambak ko taginan, then your adenovirus causes respiratory infections, mga conjunctivitis. Your picorna, the very small RNA viruses, that's right there. Picorna, pico-RNA virus, small RNA virus. If your rhinovirus causes colds, your polio, your hepatitis type A and type B, Coxsackie, no, which causes mga respiratory infections, usually in children. Your mixed viruses, important, no? They also cause your mumps. My mom's joke. Yung mga bayok, bala? That's your mumps. Your measles, rubiola. This is your typical na measles. Because there's two. There's also the German measles, which is your rubella. This one is your rubiola, caused by a paramyxovirus. And your rabies, of course, caused by a rhabdovirus. What else? Mga yellow fever, you don't have that here. And then, of course, your retrovirus, no? You have your leukemia and your AIDS. Now, sometimes your viruses will leave remnants. They're called inclusion bodies, and they're seen in infected cells, and sometimes are used as diagnostic tools. So, for example, sa brain of people who had rabies or died from rabies, because rabies is 100% mortality. Once you have rabies, you cannot get treated. There's this inclusion bodies in the brain called negri bodies. You can find this in their brain. If you do a biopsy of their brain, you will see them. There's also your AIDS inclusion bodies. There's the smallpox called Guarnieri. Then you can also see herpes. No, if you do a biopsy of body part niya may warts, you can see there nga like the herpes inclusion. Polymyelitis, you can see inclusion bodies sa mga spinal areas na. Now, other terminologies you have to remember, no? You have your latent viruses. Latent viruses, these are viruses na they keep dormant for a long while until a certain trigger deactivate siya kag ma-cause disease. No? So for example, your shingles. Your shingles is the grandson of your sulalog, of your chickenpox. They're not the same. They're not caused by the same viral entities, but your shingles, it only comes out when you're in a state when the environment allows it. So kung stressed ka, ano na. So usually it would keep dormant in your nerve cells for a long, long time. Tapos magagawa na siya kung ma-stress ka. So, that's your shingles. So, shingles is very painful. Tsura, yun nando sa lalog, pero they only choose one part of your body. It's called dermatomal. So there's only, because our bodies, we have, may mga divisions na siya of nerve bodies, no? So for example, sa, ano na siya, pa downwards na siya. So that's a dermatome. So clump lang na siya of nerves. So, amo na siyang preference sa imo shingles, dermatomal yung distribution. So for example, dili lagi na siya yung magwa, may dermatome ka na. Or isa lang ka parts ng face mo, dili na nasa sa agtang, kaya may dermatome ka na. Lain man na, di nga dermatome, lain man na. Amo siya tawag nila ka ficus. Next is your oncogenic viruses. Your oncogenic viruses are viruses that cause cancer. For example, your human papillomavirus. Certain may variants mo na sila, no? So certain variants cause cancer, cervical cancer. That's why they're oncogenic. Your hepatitis B virus is an oncogenic virus, causes liver cancer or liver cirrhosis. So what else? Your viroids and your prions, no, they were discussed earlier. So, something nice to know with prions. There is this one group of tribe somewhere in Oceania. So, mga Papua New Guinea, I don't know. Basta where cannibalism is a norm. Okay. So these people, they suffer from a disease called kuru. No? So kuru is like encephalomyelitis. So medyo, you can call them na medyo nagabuak sila bala. It's called kuru. It happens when it's very endemic in a certain tribe somewhere in Oceania because of their cannibalistic culture, eat of people. So apparently it's found out by scientists that they have a certain type of prions, or inclusion bodies in their brain that explains for their kuru. So abo na siya. Kung kuru, you look it up, there's a documentary about it. Okay, so now we move on to morphology of your bacteria. So your bacteria, they have different shapes and sizes and all that. So amo ni siya ang aton nga taxonomy sa ila, no, depending on their morphology. So this time we have it basing on their cell shapes. So you have a cocciform, no, the coccus. You have your curved or spiral shape, or anywhere in between your cocci and your bacillus. So sometimes we call this the coccobacillus sometimes. And then you have the bacilli, ano siya, elongated siya yung shape. So may certain sila nga mga description, no? Particularly certain microorganisms. Sorry guys, tuyo-tuyo ko. May tinulugan. Yung anak ko. We're sorry. Okay, so for example, your gonorrhea, nga causative agent, which is your Neisseria gonorrhoeae, they are actually cocci. So if you look at them stained on a microscope, this is what you're going to see. You will see cocci in pairs. So it's called a diplococci. A streptococci is cocci in chains. Usually your Streptococcus pyogenes. Ano na siya, no? Makita sa microscope, ano siya, chain. Strep throat ang ginakos, no? Sakit o tunlan, ba? Kung bacterial siya, Streptococcus pyogenes na siya usually. You also have your staphylococci, which is cocci in clusters or grape-like clusters, no? It's one good example is your Staphylococcus aureus, which causes boils. Yung mga katul-katul yun ang lan. There's also tetrad, no? Or a packet of 4 cocci. For example, your Microscope, rarely pathogenic. And your octad, no? Your Sarcina, rarely pathogenic. They're not medically significant. So, going back to our topic again on gram stain, this is where we can apply it now. Diba? So, diba I told you that the first step in identifying bacteria is you stain first, do a gram stain. So, once nga you have identified if the microorganism is either gram negative or gram positive, you look at its morphology. For example, okay, kung gram positive siya, if it's gram positive, does it look like a bacilli on microscope? Sa microscope mo ni makita, guys, ha? So, kung sa microscope makita mo siya bacilli siya, tatlo lang na sila. Gram-positive bacilli, tatlo lang. Can be a bacillus, it can be a bacillus Listeria, bacillus, or Corynebacteria. If it's cocci looking, you check, is it chains? Is it in chains or in pairs? Or is it in clusters? And then, going down now, kapag ina-test to further um, specify, to further identify kung ano ina siya nga microorganism. So, you can also do coagulase test, you can do a Killam test, you identify ano iyang hemolytic properties. So, amo na siya. So, now, kung gram-negative naman niya, so kung gram-negative bacteria, usually pink na siya or red, no? So, you identify, ano ang tsura yan ng microscope? Is it cocci? Is it bacilli? Or is it a cocobacilli? Kung cocobacilli, do ka somewhere between coccus and bacilli, hindi ka specify it, or somewhere in there. Now, kung cocci, so ang pinaka-common would be Neisseria, meningitis, gonorrhea. No? Kung bacilli, you check out if does it look curved or spiral, no? So if it's like that, probably Vibrio or Campylobacter. Now, how do you know kung enteric siya or non-enteric, no? You do, and actually, no, hindi magini siya ano, pero the second step would be lactose fermentation. It's a different topic for another time. But I want you to know nga, we also do lactose fermentation, we do an oxidase test. So, amo na, because to further distinguish these gram-negative bacteria. Okay, so amo na siya mag-identify. At least one of the many things we do to identify bacteria. Okay, now I want you to look at this. I know this is also in your video in your lab, but check this out anyway. This is steps to gram staining.
In this video, you will learn how to perform gram staining. This technique utilizes a 4-step staining procedure with 2 different dyes and is one of the first tests used when trying to identify unknown species of bacteria. Using a wax pencil, label a microscope slide with your initials. Then draw a circle about 1 centimeter in diameter on the slide. Using a metal inoculation loop, sterilize it by flaming. Using aseptic technique, add a drop of sterile water so that a film appears across the loop. Transfer the water into the circle drawn by the wax pencil. Flame the metal loop again to sterilize. After allowing it to cool, use the metal loop to transfer a colony of bacteria growing on an agar plate. Make sure to lightly touch the bacterial colony to avoid transferring too many bacteria. When finished, flame the loop to sterilize. Let the liquid completely evaporate, leaving behind a dry spot on the slide. This will take at least 10 minutes. Once the liquid has dried completely, heat fix the bacteria to the slide by quickly passing the slide through the flame 3 times. The flame should touch the slide for about 1 second during each pass. Ensure that all Bunsen burners are turned off before obtaining the set of gram stains. The decolorizer contains a high percentage of alcohol and is very flammable. Add a few drops of crystal violet stain to the bacteria until the wax circle is filled with stain. Let this stand for 1 minute. Rinse the slide by dipping it in a beaker of water 2 to 3 times until most of the stain has been washed off. Next, add a few drops of grams iodine to cover the bacteria, and then let this stand for 1 minute. Rinse the slide in a beaker of water until most of the iodine is gone. A second beaker of water can be used to ensure most of the iodine has been rinsed off. Now add a few drops of decolorizer. Let the decolorizer flow over the bacteria at a 45-degree angle until the flow is clear. Then finish by rinsing the slide in a beaker of water. Counterstain by adding safranin stain to cover the bacteria and let it stand for 1 minute. After 1 minute, completely rinse the slide in water. Allow the slide to dry completely. Once the slide is completely dry, observe the sample under a microscope.
No? On the microscope, may microscopy naman siya. So, kung yung tsura niya on the microscope. So, kung botak mo ng slide, no? And then, first you go sa imong low power objective, ang 10 times. And then, ang yellow, no? 40 times. Use your coarse adjustment na, bandako. Once nga na-find mo na ang imo nga focusan, this is, pero may ka times na location mo. Sorry. So, wait lang. Lunahan ko siya. Okay, this is your 1,000 times. 1,000 na dito. So, as you can see, no? Medyo purple. Sorry. So, as you can see here, no, muna yung mga cocci na siya, no? Nga ka, they look like in clusters. So, this is your gram-positive na muna siyang tsura niya. Do ka bluish purple. And then your gram-negative bacteria there at the back, as you can see, very bacillus itself. Pink na siya or red. So, muna siya na, this time, you reach it there, then you do your fine adjustment knob. Namin na kung microscope niya nga suga, hindi yellow, puti, bala, kaya makita mo, hindi na siya klaro-klaro, hindi na siya. Oh, so, there. So, round in shape cocci, rod-shaped, bacilli. So, anong arrangement niya? No? Is it in clusters, in chains, in chorba-chuchu, eklavu? You have gram-positive cocci in singles, pairs, and clusters, and then you have gram-negative bacilli nga rods. Bacilli din saya nga. Gram-negative bacilli. So, mo na siya, ha? Okay. Once makita mo na siya, hindi mo na lang malimutan. Okay? Okay, so we have other staining procedures, no? Because as I've said, simple, ang ato niya mga simple ng mga tissue staining, we can do it simply with methylene blue. Mga ano na ba yan? Mga squamous cells, yung mga cheek cells, pwede mo na siya ma-stain lang with methylene. We have a structural staining. Sabi ko to explain ko sa inyo kanina. No? You have your capsule stains, your flagella stains, and spore stains. And then kung differential, we have the gram stain. We also have the acid-fast stain. Where do we use acid-fast stain on? We use this sa Mycobacterium tuberculosis. Okay, because your acid-fast microorganisms cannot be stained with your regular gram stain, like your TB. Okay, so mo niya siya, no? Take a screenshot of this slide and familiarize yourself. Okay? Yeah. We'll get onto it in the next chapters of the lesson. Okay. Now, we also have tests for mobility. This is usual nutrient agar, no? We do a stab. So, mag-stab ka, bunon. No? So, ang imong inoculum is not on a rod, but it's on a doka palaba na siya wire. Tapos, ano, nang inoculum, that's the culture or the microorganism, and then you stab it sa imong agar. And you would know if this organism is capable of motility. Kung makita mo na, there's a grow away from the stab line. So, as you can see, sorry. Here, 1, it's a non-motile organism. Number 2 is also motile. 3 is very motile. Okay, next is this is
exactly. We'll try to put it on a. This is a non-motile bacteria. I have taken 2 bacteria. One is E. coli, straight. The motility. Let's see the experiment. This experiment is to demonstrate the motility of bacteria.
No? This is
so we have taken 2 bacteria. One is E. coli, another one is Staph aureus. So one of this is going to be motile, another one non-motile. So what we try to do, we try to take the bacteria from here, and we will prepare a bacterial suspension like this, right. So this is the organism B, and this is a organism A, right. So this A will be E. coli and B will be Staphylococcus aureus. So let's try to do the motility test. So what are the requirements? We need the bacteria in suspension, and then we need cavity slides. If you try to look into the slides, there is a small cavity here. This is a cavity. You can see the cavity. This particular angle. So you can see there is a cavity.
Wet mount or
concave
the hang
cavity.
drop niya. So dapat
Right.
slide mo.
So this is where the
organism.
drop will hang, right. Okay. And the other requirements are, you need a cover slip, you need an applicator, and you also need some paraffin wax. Right. So what we basically do, we take the paraffin wax and put it on all the corners, and we will take a suspension of the bacteria and try to place it in the center, and then we will try to hang it on the slide. Then we will try to focus these slides under the microscope, then we can see whether the bacteria moves or not visually. So this is a test which is got no ambiguity. You can see the bacteria literally moving under a microscope. Okay, let's do it. So we already prepared the bacterial suspension from the plates. For A, B is Staphylococcus aureus, and A will be E. coli. Okay, let's work with this. The first step, take the applicator stick and touch on the paraffin wax, apply a little volume on 4 corners, don't try to add too much. So you can apply little wax on 4 corners. Do not apply too much, because if you add too much, the wax can come and touch the bacteria. All right, that's all. We have applied the wax, and this is what is required now. All right. The next step is that I take the bacteria. So I take the bacteria A and B. First, let me take the bacteria A. Right. So first, we will take the suspension A, I take a small drop and place it in the center of the slide. It's already been placed there. You can see the drop is placed. And then I'll take the bacteria B and place another drop. Okay. Try to keep the drop as small as possible. If you keep a bigger drop, it may not remain as a drop. It can disperse off, then the whole experiment doesn't work. So now we have the important part. I'm going to take the A and try to invert it onto the slide and gently press. That's it. So you can see the drop hanging from the slide. This should not be disturbed. We take this undisturbed to the microscope and do an observation. In the same way, we'll also check, for the drop B. Take the slide, invert it, and place it on the cover slip. Now you can see the 4 corners are stuck with the paraffin wax, and slowly remove it and place it like this. So now both the slides are ready. We'll try to observe it under the microscope. Let's do the microscopy now. Thank you for listening.
So, no, if you do a microscopy, you can see Motility bacteria. Hindi, ma-distinguish mo siya kay compared to like just regular brown yan, yung movement, yung ado ka random, ang ilay, ito ka, yeah, like it's sila yung trajectory. It is trying to go somewhere. So that is your Motility bacteria. Now, with your non-motile bacteria, such as your Staphylococcus aureus, wala movement. Staph aureus is not motile. Now, this one naman niya is an example of a parasite called Trichomonas vaginalis. Sorry, that's my cat attacking my chair right now. I'm so sorry, guys. So this is an example of a wet mount. So where can you find Trichomonas vaginalis? No? Usually, sa mga urine samples, may gaupod yung Trichomonas. Trichomonas is a sexually transmitted disease. It causes trichomoniasis. So, amo na siya ho. You can see there. That's what it is. It's trying to go somewhere. Hindi siya kasi nga do brown yan. And when you say brown yan, it's like duga ka diffuse lang siya. This one, you can see it's tail. It's trying to move around. So, okay, that's your trichomonas right there. Here. Why be responsible adults? Okay, para you don't have things like trichomonas. Okay, so these are examples of sa ano naman niya, colonies growing on agar. You can actually figure out what a certain microorganism is basing on its behavior on a particular agar. So for example, this is on your upper left. This is your Serratia marcescens. No? You know it's Serratia because it assumes very red colonies. So, muna siya. Serratia usually, mula yung medically significant na red ang colonies. The one here in the middle, this is your E. coli or your Escherichia coli. This is your Mac-Conky agar. You would know this is E. coli on if it's on Mac-Conky because, well, you can't really know it's E. coli. What you can actually surmise from this is that this is a lactose fermenting organism. Why do you say it's lactose fermenting? McConkey agar has lactose in it. So once that a microorganism is lactose fermenting, it assumes the color pink. So this is a lactose fermenting organism, most likely E. coli, because E. coli is a lactose fermenting microorganism. Here on the right, this is also actually E. coli. This is your EMB agar. Your E. coli is most specific on your EMB agar. Why is it? Because it produces this blue-green metallic sheen. No, that's why you would know nga ang E. coli, not because it's EMB agar kung may metallic na bluish-green sheen. This one here in the bottom left, as I think I'm trying to say earlier, this is your streptomoniae, no? As you can see on the blood agar, you can find slimy, very slimy gid. No, that's the capsule. So, as you can see, mo naging inoculate niyo, sobra niya ka-inoculate, kinararo niya, tama ka duot sa iya nga inoculation sa agar. Dapat di mo na pagduoton. Napaka-smooth lang. Here in the bottom, this is hemolysis patterns. This is a blood agar. And certain gram-positive microorganisms have a particular hemolytic pattern. For example, here in A, this is beta hemolysis. No? Some gram-positive microorganisms are capable of beta hemolysis. We call this beta because this is yellow. The one here in the middle, it's green. This is alpha hemolysis. And then see here, doon ka medyo transparent lang siya, doon ka wala color. That's your gamma hemolysis. So, na siya, certain microorganisms, I think, certain, So, for example, here, is this a hemolytic pattern? No. Ari lang. So, if the hemolytic pattern on the blood agar is beta or yellow siya, you can probably tell it, it's strep, pero it's probably group A strep, group B strep, group C, group G. Kung alpha ang pattern of hemolysis, it's probably viridant strep. If it's gamma pattern, it's your enterococci. Ano siya? Your kelang test is a test for capsule. So, muna S pneumoniae positive siya. Or if you culture it on an agar, ano na siya? Very slimy ang iyang colonies. And then here on the lowermost right, this is your Protists vulgaris or mirabilis on a blood agar. How do you know this is Protists? Because it's swarming, it swarms. Regardless of your inoculating pattern, sa pabi na, di ba? Kung nag-inoculate ka, go-follow siya sa pattern. Ini saya swarming. So wala siya gapate sa alagyan. It goes its own way kag it will invade the entire thing. That's how Protists is. So also remember that your bacteria only grows with certain requirements. No? Medyo choosy sila. So you would also know what sort of microorganism it is. You can identify it with the kind of relationship to oxygen it has. So for example, you have obligate aerobes, microaerophils, facultative anaerobes, aerotolerant aerobes, and obligate anaerobes. And you're like, what? What is this? So obligate aerobes need oxygen all the time. Okay? Microaerophils, gamay lang nga oxygen ila kinahanglan. Facultative anaerobes, they need a very small amount of oxygen, but mostly wala oxygen. Aerotolerant, they're like, okay lang kung may oxygen, pero we won't thrive on it, but it's fine, we can deal with it. Obligate anaerobes, no oxygen. It's like, I cannot grow if there's oxygen. Okay. So, for example, this is your thioglycolate broth. Okay. So, obligate aerobes will grow sa pinaka-topmost part of this tube, wherein there is 20 to 21% dissolved oxygen. You'd be wondering, nga 20 to 21% lang na how? Because room air temperature room air oxygen, you know, the oxygen that we breathe in right now sa ating atmosphere, it's actually 21% dissolved oxygen. Oxygen is actually pretty toxic. If you go up to 100%, mas mapatay kasi na. So, kita ya in room air, as long as normal kita, pag wala kita ya special requirements like kapukon ka, or may COVID ka, or yung tubuhan ka, we thrive in 21% oxygen. And so do obligate aerobes. Now, microaerophils will grow at around where there is 5% oxygen. So usually sa may mga bottom parts na. But obligate anaerobes, they will grow where there is no oxygen. So sa pinaka-bottom sila usually. Okay, now familiarize yourself with this. Okay, take a screenshot and try to familiarize yourself. Okay. Special. Rickettsia, we don't have that here actually. Ehrlichia, we don't have it here. Anaplasma. Chlamydia is very common actually in the Philippines. Chlamydophila pneumoniae can happen. Psittacosis, not really. Mycoplasma pneumoniae can happen. And some non-gonococcal urethritis. Mga specialist ang mga UTI. Okay, mycoplasma kag ureaplasma. Ang imong scrub typhus or your Orientia, we don't have that here. Okay, now we move on to your Archaea. Now this is a bit long, pero you know, try watching it anyway, okay?
We've spent the past few months talking about animals here on Crash Course, specifically human animals, because humans, we love talking about ourselves, and also because animals are just really interesting. But it's high time that we talked about the rest of the living world. Because I hate to break it to you, but most of the alive things on Earth are single-celled organisms. And by most of the alive things, I mean that these organisms make up 2 of the 3 taxonomic domains of all life, plus 1 of the 4 kingdoms. I'm talking about Archaea, Bacteria, and Protists. With the exception of a few Protists, they're all unicellular, and they are by far the most abundant and diverse organisms on Earth. Maybe more importantly, they lay claim to the world's oldest and earliest living lineages, dating back to the very first twinkle of life on this planet. So by understanding these 3 groups, you begin to truly understand life on Earth, its origins, and how everything that came after them, including us, came to be. What's more, because their heritage is so ancient, these organisms often take weird, cool forms that don't look like life as we think about it, and they do amazing things. Some not only live but thrive in environments that would kill you, me, and everything we hold dear, and others make their living by invading organisms, including us, and causing disease. Then there are those that do the opposite, making life possible by fixing nitrogen from the atmosphere, helping animals digest food. Members of these groups have names like sailor's eyeballs and dog vomit slime mold, and they can take the shape of rods, blobs, corkscrews, or coils. Kinda like the doddering, eccentric relatives you're forced to spend some holiday with once a year, the Archaea, Bacteria, and Protists are our oldest, oddest relatives. And it's about time you got to know them. There's no denying it. Every multicellular organism on this planet, whether it be a mushroom or a vampire bat, evolved from a single-celled organism. And while some of these single-celled organisms evolved to populate the world as rhinos and strangler figs, others found happiness in the unicellular lifestyle, and they haven't changed much in the past few billion years. Today, nearly all unicellular organisms are either Archaea, Bacteria, or Protists. Protists, you'll recall, are eukaryotic organisms that make up the kingdom Protista under the domain Eukarya. Bacteria and Archaea, meanwhile, are their own prokaryotic domains. And I hope you haven't forgotten this. The big difference between prokaryotes and eukaryotes is that eukaryotic organisms, including you and the plants and fungi and animals that you know, have cells with a nucleus that hold their genetic information, while prokaryotic cells don't have a nucleus or any organelles to speak of. These 2 groups do have some important things in common, like having plasma membranes that are filled with cytoplasm and ribosomes that contain RNA and synthesize proteins, and they both have DNA that carries the instructions for operating the cell. But eukaryotic DNA comes in strands in the form of chromosomes, while prokaryotic DNA is found in rings called plasmids. So again, and this time with feeling, Protists are mostly single-celled eukaryotic organisms. Archaea and bacteria are single-celled prokaryotic organisms. The word prokaryote actually means before the nucleus, which is a clue that prokaryotes are an older form of life. And we literally cannot find anything older than Archaea. The first Archaea fossils date back to 3.5 billion years ago. I'm talking just a billion years after the Earth formed and was still bombarded by comets and meteors, not to mention fried by UV radiation. But in the midst of all that, Archaea or just chillaxing. Earth's climate has calmed down since then, so today Archaea are found in some of the world's most extreme environments, in underwater hydrothermal vents, in oil wells, and volcanic hot springs, even acidic mine drainage. Archaea were probably the earliest living things, and their adaptability is probably what allowed them to take root in Earth's early kind of grody environment. One key group of the Archaea are the methanogens. These guys perform more moderate environments like mud, swamps, and your intestines, but they derive energy from hydrogen gas and carbon dioxide, which is pretty cool, and they emit methane as their waste product. Methanogens, methane generators. We know that waste as swamp gas, and also other kinds of gas. The other groups are extremophiles, which not only tolerate but prefer really wicked surroundings. The most famous of these are the thermophiles, which live in temperatures that would melt your face off. I mean, serious. Pyrolobus fumarii, a species of Archaea discovered in the late 1990s in a hydrothermal vent, live at temperatures around 113 degrees Celsius. Not Fahrenheit, Celsius, significantly above the boiling point of water. Most organisms can't take heat like that because it causes their DNA to unwind and their proteins to denature or permanently change shape, but thermophiles have evolved adaptations that keep them stable at these screaming hot temperatures. There are also halophiles, or salt lovers, which live in places like the Dead Sea or the Great Salt Lake, probably Daniel Tosh's mouth. Most halophiles breathe oxygen and are heterotrophic, but there are some bizarro outliers, like species that use sunlight to make energy, but not like plants do. They have light-harvesting pigments in their membranes that react with light and enable the cell to make ATP for energy. I know, it's crazy, but despite their alien-sounding ways of life, Archaea really aren't all that different from bacteria, which are also prokaryotes. In fact, Archaea and bacteria were classified together for much of the 20th century. It was only when scientists realized that they had some important genetic differences, like in the sequence of their ribosomal DNA and the makeup of their RNA, that they were separated into two domains. Bacteria are nearly as ancient as Archaea. Fossils show that they were widespread about 1.5 billion years ago, but there's evidence that they've been around for more than 3 billion years. Today, they make up the vast majority of prokaryotes on Earth, and they're super slick when it comes to adapting quickly. Many bacteria are parasitic. Think your strep throat, your staph infection, anything you've ever taken an antibiotic for. But bacteria can fend off antibiotics and the ninjas in your immune system by garbling up their DNA from one generation to another. They can randomly turn genes on and off, creating unique genetic combination as its population multiplies, keeping its host immune system and drug makers on their toes. Like Archaea, bacteria don't reproduce sexually, but bacteria have devised a way to pass their genetic material to their buddies, a little trick called horizontal gene transfer. For example, you've heard of antibiotic resistance, right? Well, horizontal gene transfer is one reason for it. A strain of bacteria that has genetic resistance to an antibiotic can pass some of its DNA and that drug resistance to another strain, which is why we're always in kind of an arms race with the bacteria of the world. And of course, bacteria are incredibly diverse, with too many phyla to name, more than 24. But one way of classifying them is by their different kinds of cell membranes, which react differently to a staining technique scientists use called gram staining. Gram-positive bacteria have thick cell membranes, and they're a huge group that includes species that live individually, like Staphylococcus and Streptococcus, as well as some colonial bacteria that are responsible for diseases like leprosy and tuberculosis. And there are lots of gram-negative bacteria too, which have thinner membranes. The biggest group here are the proteobacteria, named after Proteus, because they take so many forms. These include bacteria that make our lives possible by converting nitrogen in the atmosphere to compounds available to plants, as well as others that cause stuff like food poisoning and Legionnaire's disease. Cyanobacteria, meanwhile, are the only prokaryotes that use photosynthesis to make their food, and they're some of the most important members of aquatic food webs, providing microscopic forage for all kinds of freshwater and marine ecosystems. Spirochetes are the corkscrew-shaped bacteria that you've no doubt heard of. Most are harmless, but a couple of parasitic species are the culprits culprits behind illnesses like Lyme disease and syphilis. And speaking of sexually transmitted diseases, the last major group of bacteria worth mentioning are chlamydias, which are strictly parasitic and live only in animal cells. They're scumbags, obviously, and are the leading infectious cause of blindness in the world, as well as that eponymous infection of the urethra that makes me kind of want to cross my legs just thinking about it. So Archaea have managed to make a nice multi-billion-year living by surviving in weird, out-of-the-way places, and bacteria have developed ways to pass their DNA without sexual reproduction. But you know who's a hot freaking mess? Protists. Evolutionarily, they're the youngest of the three, having evolved from bacteria around 1.7 billion years ago, and in a lot of ways they're more sophisticated. For starters, they're eukaryotic, but also some are multicellular, and a few kinds can even reproduce sexually. But their domain is a big crap circus because some protists seem to be more closely related to plants or animals or fungi than other protists. So scientists tend to talk about them based on what else they resemble. There are protozoa, which are kind of animal-like, algae, which are kind of plant-like, and fungus-like ones, including the tastefully named slime molds. The one thing all of these have in common is they need to live somewhere wet, in a bog, or in your body, or in a snowbank, wherever. Protozoa are actually really cool because they're like tiny animals. Like us, they're heterotrophs, so they have to eat other stuff in order to live, and because they need to eat, they've got like mouth parts, or at least mouth part sorts of things. And they can move around by using all kinds of really cool structures. Some have flagella, the whip-like tails to propel them through the water, or cilia, little hair-like structures that work like oars, and some move around with a kind of blobby amoeba-like motion. I say amoeba-like because the protozoans that move this way are amoebas. And speaking of amoebas, some protozoans are parasitic. You've probably heard of amoebic dysentery. That's caused by amoebas. Malaria is caused by this little guy, a protozoan called Plasmodium vivax, while African sleeping sickness is caused by Trypanosoma brucei, this guy here. Moving on to the plant-like protists, which are algae. All algae photosynthesize like plants, even though they're not plants, because they use different kinds of chlorophyll molecules. Some are unicellular, like tiny diatoms, which have a hard shell made of silica. The amazing thing about single-celled algae is that they can get really honkin' huge. For example, ladies and gentlemen, cast your gaze upon the sailor's eyeball, thought to be the biggest single-celled organism on the planet, also known as bubble algae. It lives on the seafloor in tropical oceans and can grow up to 5 centimeters across. How is that thing one cell? Anyway, you already know multicellular types of algae, aka seaweed. They're closely related to land plants, as you can tell by looking at them, and they're generally grouped into red, green, and brown varieties, although these all have their unicellular forms as well. The green algae are probably what gave rise to land plants about 475 million years ago. They're the most abundant and diverse, and they have chloroplasts very much like land plants. So they can only live in shallow water because they need a lot of sunlight. Red algae is able to live at greater depths and has an extra pigment in it called phycoerythrin, which gives its chlorophyll a boost in deeper waters. And brown algae is what most of the seaweed you see in the ocean is. Kelp is an example. They're the largest and most complex of the multicellular algae. Finally, we have our fungus-like protists, which include the delightful slime molds. They absorb nutrients from their environment and produce fruiting bodies like fungi. But even though they look like piles of barf, they can actually move around like an amoeba and eat bacteria by phagocytosis. Slime molds can be pretty easy to spot because they're often brightly colored, like this charming species, which in all seriousness is known as dog vomit slime mold. You heard me, these organisms are so freaking screwed up that scientists couldn't think of a better name for it than dog vomit slime mold. Like I said, they're old, they're odd. Get used to it. Thanks for watching this episode of Crash Course Biology. If you want to catch up on anything that you're a little fuzzy on, table of contents over there. Thanks, of course, to all the people who helped put this episode together, and if you have any questions for us, please, Facebook, Twitter, or the comments below. Goodbye.
So your protozoa are mostly single-celled free-living organisms. They have eukaryotic organelles, so they cannot photosynthesize. So your protozoa have two typical life stages. They are either trophozoite, this is their feeding stage, and they can also be in their cyst stage, which is their dormant stage. So if the environment is friendly and they can feed, they resume their trophozoite stage. And when it's unhealthy for them and they think they cannot survive, it's their cyst stage. So some protozoans are parasitic. No, they cause diseases, and some are mutually symbiotic. So, you familiarize yourself with this, huh? Okay. So, for example, isa na lang ka-example. Your amoeba, no? One of your example of your amoeba is your entamoeba histolytica, no? This is a common cause of your amoebiasis, no? Yung gina-amoeba ka wala. So, when you do a stool exam, this is where we base kung anong stage ang amoeba sa imo, if you have the trophozoite or the cyst stage. So that's why we give 2 medications for our patients suffering from amoebiasis. Kaya dapat i-treat mo ang trophozoite stage kag ang cyst stage. You cannot just choose one. Dapat mapili. Both dapat imong pull mo. Okay, so other medically significant protozoa, no? So, amutamba ko sa inyo ang entamoeba histolytica. This is your causative agent for your amoebiasis and amoebic abscesses, mga abscess ng balas ng atay, caused by amoeba. That's your entamoeba histolytica. There's also your Naegleria fowleri, which causes amoebic meningoencephalitis. And then you also have your Acanthamoeba species. It's not common sa ato, no? But Naegleria, muna siya makita mo siya sa mga ponds, and then they go inside your ear, tapos susunod yung brain, and then they will cause amoebic meningoencephalitis. Now you also have your ciliates, no? Okay, balik sa amoeba, no? Your amoeba, they have pseudopods, right? Your ciliates have cilia. So, ang ilan niya nga movement is oar-like. Nung naman siya bago sakit ka sa boat, tapos ng do oar na, oar-like motion. Your medically significant ciliated protozoan is your B. coli or your Balantidium coli. Causes dysentery. Another example are your flagellates, no? Ang ilan nga movement is wave-like. So, another example is your Trypanosoma species, your trypanosomes. They cause, for example, your Trypanosoma brucei gamiense caused by the vector, meaning ano ang ginasakyan niya nga insect, is your tsetse fly. It causes African sleeping sickness. You also have your Trypanosoma cruzi that causes Chagas disease. You also have your trichomonas, no? Your trichomonas, vaginalis. And then you also have your trichomonas vaginalis. Sorry, that's not trichomonas, it's trichomonas. And then you also have your Giardia lamblia that causes giardiasis. You also have non-motile protozoa, no? So, example of this is your sporozoites or your sporozoa. And the best example for this is your Plasmodium species that causes malaria. So you have Plasmodium vivax, mga Plasmodium ovale, damo na sila. Plasmodium falciparum, and then their vector, Anopheles, and they cause malaria. Very endemic in Palawan. Other sporozoites, you have your Cryptosporidium, Babesia that causes babesiosis, your Cyclospora belliae, and your Toxoplasma gondii from rats. So now we move on to your fungi.
Hello and welcome to the wonderful world of fungi, or fungi. Both are acceptable pronunciations, but I say fungi because it's fungus, not fungus, though fungus is also fun to say. Fungi are a little bit like plants and more like animals than you might think. They diverged from protists about 1 billion years ago, and today scientists estimate that there are about 1.5 million species of fungi on the Earth. Though in a formal taxonomic way, we only know about 100,000 or so of them. And those that we have met are wonderful, weird, and in some cases deadly. The fact is, death is pretty much what fungi are all about. Sure, there are the fun fungi, like the single-celled Saccharomyces, also known as yeast. Without them, we wouldn't have beer or wine or bread. It's also true that fungi are responsible for all kinds of diseases, from athlete's foot to potentially deadly histoplasmosis, aka spelunker's lung, caused by fungus found in bird and bat droppings. Fungi can even make people crazy. When the fungus Claviceps purpurea grows on grains used to make bread or beer, it causes gangrene, nervous spasms, burning sensations, hallucinations, and temporary insanity. One compound in this fungus, lysergic acid, is the raw material used to make LSD. And finally, there's the destruction that some fungi bring onto other animals. More than 6 million bats in North America have died since just 2007 due to a fungal disease called white nose syndrome. And a fungus has been implicated in several extinctions of amphibians and threatens many more, perhaps as many as a third of all amphibians on Earth. But none of this is what I mean when I talk about fungi and death. While some members of the fungus family are total bummers, all of them together perform perhaps the most vital function in the global food web. They feast on the deceased remains of almost all organisms on the planet. And by doing that, they convert the organic matter that we're all made of back into soil from which new life will spring. So fungi, they thrive on death, and in the process, make all life possible. Aha! You didn't expect to see me in the chair so soon, but before we go any deeper into the kingdom of fungi, I wanted to make a toast to Louis Pasteur in the form of a biography. By Pasteur's time, beer had been brewed for thousands of years in cultures all over the world. Some experts think it may have been the very reason that our hunter-gatherer ancestors started farming and cobbled together civilization in the first place. But for all those millennia, no one understood how its most important ingredient worked. Until brewers could actually see what yeast were doing, the magic of fermentation was essentially magic. Pasteur himself was never a big beer drinker, but part of his academic duties in France required him to help find solutions to problems for the local alcohol industry. And as part of this work in 1857, he began studying yeast under a microscope and discovered that they were, in fact, living organisms. In a series of experiments on the newfound creatures, he found that in the absence of free oxygen, yeast were able to obtain energy by decomposing substances that contained oxygen. We now know that Pasteur was observing yeast undergoing the process of anaerobic respiration, aka fermentation, breaking down the sugars in grains like malted barley and converting them into alcohol, carbon dioxide, and the range of flavors that we associate with beer. Along the way, Pasteur also discovered that beer was often contaminated by other bacteria and fungi. The growth of these beer-spoiling microbes, he found, could be thwarted for up to 90 days by keeping the beer between 55 and 60 degrees Celsius for a short period of time. Today, we call that heating process pasteurization, and it's used in everything from milk to canned foods to syrups to wines. For our purposes, the thing to hold on to here is Pasteur discovered that yeasts decompose sugars to get energy. And it turns out, most fungi spend most of their time decomposing all kinds of organic matter. Often the matter is dead when fungi get to it, but not always. When a tree, or a person, or a deer keels over, fungi move in and start the work of decomposition. Same goes for that orange you forgot at the bottom of the fruit bowl. If it weren't for this fungal function, plants and the animals that eat them couldn't exist, because the elements that they take from the soil would never return. Thankfully, the decomposition performed by fungi recycles the nutrients for the enjoyment of plants and animals, as well as for other fungi. All of this points to one of the main traits that all fungi have in common. From single-celled yeast to giant multicellular mushrooms, fungi, like us, are heterotrophs. But instead of eating, they absorb nutrition from their surroundings. They do this mostly by secreting powerful enzymes that break down complex molecules into smaller organic compounds, which they use to feed, grow, and reproduce. Most multicellular fungi contain networks of tiny tubular filaments called hyphae that grow through and within whatever they're feasting on. Unlike plant cell walls, which are made of cellulose, the cell walls of fungi are strengthened by the nitrogenous carbohydrate chitin, the same material found in the exoskeletons of insects, spiders, and other arthropods. The interwoven mass of hyphae that grows into the food source is called the mycelium, and it's structured to maximize its surface area, which, as we've learned in both plants and animals, is the name of the game when it comes to absorbing stuff. Mycelia are so densely packed that 1 cubic centimeter of rich soil can contain enough hyphae to stretch out 1 kilometer if you laid them end to end. So as hyphae secrete the digestive enzymes, fungi use the food to synthesize more proteins, and the hyphae continue to grow, allowing the fungi to conquer new territory and grow even more. As a result, fungi can get crazy big. Record-holding big. A single honey mushroom in the Blue Mountains of Oregon is thought to occupy some 2,386 acres. by area the largest organism on the planet. Now, there are all kinds of crazy ways that fungi are classified, but probably the easiest and most useful is organizing them by how they interact with other organisms. The straight-up decomposers that break down dead stuff, the mutualists, which form beneficial relationships with other organisms, especially plants, and then there are the predators and the parasites. Decomposer fungi secrete enzymes that break down and absorb nutrients from non-living organic material, such as that tree that nobody heard fall in the forest. In fact, the ability of fungi to break down lignin, which is like what makes wood woody, and break it into glucose and other simple sugars is crucial for the cycle of life. They're pretty much the only organism that can do that. They can even decompose proteins into component amino acids. Basically, all the black bits in the soil in your backyard are tiny fragments of former plants digested by fungi. Mutualist fungi are a smaller group. Many have specialized hyphae called haustoria that tangle themselves with plant roots for the benefit of both organisms. These guys help plants absorb nutrients, especially phosphates, by breaking them down more efficiently than the roots can themselves. In turn, the fungi send out their hyphae into the plant's root tissue and withdraws like a finder's fee, basically, in the form of energy-rich sugars. These mutualistic relationships are known as mycorrhizae, from the Greek words mykes, or fungus, and rhizon, or root. Mycorrhizae are enormously important in natural ecosystems as well as in agriculture. Almost all vascular plants, in fact, have fungi attached to their roots and rely on them for essential nutrients. Growers of barley, the main ingredient in beer, will even inoculate barley seed beds with specific mycorrhizal fungi to help promote growth. Other fungi aren't nearly so kind to their hosts. Predatory fungi actively capture prey with their hyphae. The soil fungus Arthrobotrys uses modified hoops on its filaments to snare nematodes and absorb their inner tissue. Then there are the parasites, those fungi that feed on living organisms without killing them, at least for a while. Take one of my personal favorites, the zombie ant fungus, or Ophiccordyceps. It shoots spores into an ant where their hyphae grow into its body and absorb nutrients from non-essential ant organs. When the fungus is ready to reproduce, it invades the ant's brain and directs it to march to a cool, moist location in the forest where its so-called fruiting spores erupt through the ant's head to spread even more spores. And just to prove that even fungi have superheroes, in 2012, scientists discovered that these zombie spores have themselves been targeted by another parasitic fungus. Not a lot is known about this ant-saving fungus other than it sterilizes many of the zombie spores through a process likened to chemical castration. That is so messed up. Weird. Alright, now since I brought that up, we should talk briefly about fungus sex. Fungi reproduce any way they can, either sexually or asexually. Some species even do it both ways. But whichever way they choose, most propagate themselves by producing enormous numbers of spores, much like we saw in non-vascular plants and the simplest vascular plants, the ferns. But, and this is a big but, sexual reproduction in fungi isn't like sex in any other organism we've studied so far. The concepts of male and female don't apply here at all. Some fungi reproduce on their own, others can reproduce with any other individual that happens to be around, and still others can only mate with a member of a different so-called mating type. They're not different sexes, they just have different molecular mechanisms that either make them compatible or not. Sometimes these types are called plus and minus, and other times 1 and 2. In any case, it's still considered sexual reproduction because each parent contributes genetic information when they make with the spore making. It all starts with this beautiful chemical mating dance as the mycelium from one fungus sends out pheromones that are picked up and bound to receptors by another willing and able partner. This binding compels each mycelium to send its hyphae toward the other. When they meet, they fuse the cytoplasm of their cells, a stage of reproduction called plasmogamy. Sometime between hours and centuries later, yes, it can literally take hundreds of years for fungi to have sex, this union leads to the production of spores that each fungus is then able to disperse. Certain types of fungi, including the tasty morel, produce spores in sac-like asci contained in fruiting bodies known as ascocarps. That is the part that you pick when you're wandering through the forest. Some fungi shoot their spores off into the breeze, others spores float away on the water. More enterprising spores will hitch a ride on passing critters, hopefully to be dropped off somewhere where there's plenty of nutrients to absorb so they too can grow, send out sexual pheromones when their time comes, and let their hyphae do the tango. Finally, for some fungi, sexual reproduction just isn't all it's cracked up to be. They'd rather just get on with it themselves. Some of these grow filamentous structures that produce spores by mitosis. These structures are visible and they're called molds, the stuff on the orange in the bottom of the fruit bowl or on the heel of the piece of bread that you left for a roommate who decided to leave it for the other roommate who thought that you'd rather have it. In the unicellular yeast, the asexual reproduction occurs by old-fashioned cell division or the formation of buds that that get pinched off into separate organisms. Since some species of yeast, like our beer-making friend Saccharomyces cerevisiae, convert sugars into alcohol, brewers create conditions that encourage high rates of yeast production, like giving them lots of sugar and oxygen, since more yeast means more alcohol. So yeah, fungi, they feast on death and they can make us go insane and turn ants into unholy zombies of the night, but because of their hard work in strange ways, they make possible stuff like agriculture and beer and everything else worth living for. So thanks to the fungus. And also thanks to you for watching this episode of Crash Course Biology, and of course, thanks to the people who helped me put it together. They're awesome. Thank you guys. There's a table of contents over there if you want to click on it and go review any of the stuff.
Okay, so no, these are your fungal colonies, no, and they are usually classified according to your hyphae. So amo na siya. So if your fungi or your fungi presents with a septated na hypha na may utututut, is called a septate hypha. Then you also have aseptate hypha, no? Some of your fungi are aseptate. Sometimes the yeast colonies would be like this. Sometimes they would have mold colonies. They would present with an aerial hyphae, and then they have vegetative hyphae, the roots underwater. So that's why, for example, when your bread has molds, you don't eat it, even if you see isa lang kaparting green, because it has its hypha in the entire bread. You just cannot see it. So don't eat molded bread with molds. Okay, so how do fungi reproduce, no? They can produce sexually or asexually through spores. So sexual spores, this is where they fuse to gametes. You can see this in your ascospores, basidiospores, zygospores. And then there's also asexual spores. They're called the conidia. And this is not the fusion of two gametes. But basically, what the book described is that there are so many other asexual ways that fungi can reproduce, but it's definitely not the fusion of two gametes. Amo lang na siyang point. So familiarize yourself with this, no? These are the more common phyla of fungi and type of spore that they have, and then kung asexual sa anong meron. So now your yeasts now, they lack mycelia, they have a fake hypha or a pseudo hypha, and you can visualize yeast through direct visualization or staining, and then you can see that you can visualize them on a microscope. There are special stains for yeast. Molds, penicillium sa mold. Wala naman bago. Your penicillin, or penicillium, no, the origin of your penicillin, it was actually a mold. Discovered by Alexander Fleming. Okay, so let's move on to your medically significant fungi, no? Mycosis, this is your term for fungal infections. You have superficial and cutaneous and subcutaneous and systemic mycosis. So your superficial mycosis are your dermatophytes, tinea, and some of your candida. Tinea, no, these are your ringworm. Wala na the worm sa ringworm. It's fungi. Okay, so your tinea, it would have a specific name depending kung di ng ringworm mo nagtubo. So if you have a tinea sa imo ulo, that's a tinea capitis. If it's sa imo tiil, like your athlete's foot, that's your tinea pedis. It's imo hita, no, that's your tinea curis. So more deadlier types or severe types, you have your subcutaneous and systemic mycosis. Usually you will see this in immunocompromised patients. When you say immunocompromised, that means their immune system is not working as it should be. For example, our AIDS patients. Okay. So we also have dimorphic fungi. Dimorphic fungi, muna sila ang mga, they look like yeast, or they are in yeast form. They're in body temperature, so around 36 or 37 degrees. Pero when they're not in body temp, they're molds. So these are some of these are your histoplasma capsulatum. So for example, histoplasma exists as a mold outside of the body. So sa earth, sa plants, sa mold. Where once it's in the body, it becomes a yeast, and that's when it becomes opportunistic. Same thing with your sporothrix, your coccidioides, and your blastomyces. Okay, So that's it. That's the end of my lecture. No, actually, this is just a video. This is not really an assignment. You can just watch this. Try looking at it. It's actually a video about fungi. It's very interesting. For my other sources, just Google Professor Dave Explains and Crash Course Biology. Kung gusto nyo nga, muna na lang tawon yun, you will not look at my beautiful face. Okay, so thank you guys, and I hope you learned something from this lecture, and see you next time.